We finally made it through the chem review stuff. So now we can finally start talking about cells, which is probably why you enrolled in this class in the first place. It's because you wanted to learn more about like how living things are working here. I'm going to do a little review on diffusion, just because in grading, going over those pre-labs for osmosis, a number, I'm looking here, about half of them look good, and about a half of them seemed like they were a little upside down around on what actually drives diffusion. So this is going to be helpful for those of you who I sent a little message, like please like revise and resubmit your hypothesis. This will help you out. So let's, and remember, osmosis is just a specific form of diffusion, right? It's the diffusion of water across a semi-permeable membrane. So anything we talk about that is relating to the rate of diffusion is also relating to the rate of osmosis. So if I'm thinking about, I have stuff moving from one side to another. And I'm curious about the rate, like how much stuff is moving per unit time. So usually we use this little alpha thing as proportional to. So rate is proportional to what? What things are going to affect the rate of diffusion?
Surface area.
One is going to be the surface area.
The concentration gradient.
Let me actually start writing this down here. So surface area, the greater the surface area, the more stuff is going to be able to all cross over in a certain amount of time. And we're going to see this really dramatically, like in the lining of the intestines. You're going to have all this crazy folding of the intestinal lining cells. So you end up getting a surface area the size of a football field. So you're really bringing nutrients across from your intestinal lumen into your body. We'll see this in the lungs, like all of the little alveoli, tiny little balloons branching and branching and branching to get a surface area like the size of a like tennis court inside your chest cavity there for gas exchange. So increasing surface area, and again, I'm putting this on the top here, meaning it's directly proportional. The higher the surface area, the faster the rate. Okay, then I just heard another really important one.
Concentration gradient.
Yeah, concentration gradient. That's also up on the top here. The higher the differences between the two sides in concentration, and gradient means the difference. The larger the difference, the faster the rate. A bunch of you guys got that opposite in your hypotheses, actually, in your pre-lab, right? If you have a large difference, there's going to be a much faster movement. If they're very similar, it's going to be slower. If they're exactly the same, there's no difference, there'll be no net movement. There'll be no diffusion.
So the concentration gradient specifically means the difference in water?
No, it just means the difference in whatever.
Any molecule?
Yeah, concentration gradient. It's whatever particular thing you're looking at for moving. So concentration gradient, again, in our lab today, if you have 40% sucrose inside the bag and distilled water outside versus 20% sucrose in the bag, which one of these bags is going to have a higher concentration gradient across the membrane?
The 40%.
40%.
Yeah, the 40%. The difference between 0 and 40 is way higher than the difference between 0 and 20. It's twice as large of a gradient. So which one of these cases are we going to expect a faster diffusion rate or faster osmosis rate in this case? Yeah, exactly. So kind of you want to keep that straight. What other things affect rate of diffusion?
Temperature.
Say it one more time.
Temperature.
Temperature, absolutely.
The higher the temperature, the faster the rate of diffusion.
Exactly. Yeah, because remember, temperature is just that intrinsic energy that the molecules have. So if there's higher temperature, they're just all moving faster, jiggling faster. So temperature, higher temperature is going to also be positively correlated with a faster diffusion rate. What other things? If you read ahead your lab for today, the diffusion, the demos of diffusion, you'll know.
The permeability of the membrane.
So that is in here, although that's not what I was looking for, but permeability of the membrane is also going to be here.
Isn't it molecular weight or something like that to do with weight?
Yeah, the molecular weight is going to be the last one. That's going to be on the bottom. So molecular weight is going to be inversely related to the rate of diffusion. You know, and this one is actually, it's kind of intuitive if you think about momentum. I'll illustrate it in just a moment. But this relationship here, you should look at each of these parts and make sure each one kind of feels intuitive to you. Higher concentration gradient, higher rate. More surface area to move across, higher rate. Higher temperature, a higher rate. Greater permeability, meaning it's easier to move through this like membrane, higher rate. Higher molecular weight of the things that are moving, that's going to be a lower rate. The reason we have this inverse relationship here, think about, you know, a certain amount of energy. Like I have a finger flick of energy. You know, it's a certain amount of energy. I flick my fingers. In fact, this will be easier if I just turn off my video here. So if I have, you know, something heavy, I need something heavy. Here's something heavy, like some, you know, big can, and I give it a finger flick of energy, it moves a bit. You know, it definitely moves, but it doesn't move all that much. But it has, that finger flick of energy was enough to make it jiggle a little. If I take something else, let's say I, you know, take a, here's my little highlighter. I'm going to give it a finger flick of energy. Whoa, it actually went sailing through the air and moved much farther and faster. So the same amount of energy, this little finger flick of energy with something massive, it didn't move it all that far and fast. The same amount of energy in this pen, which I'm not going to find right now because it's under the desk, made it move much farther and faster. So hopefully this is giving you some sense of this idea. Again, if I go, if we go and think about molecular weight here, the higher the molecular weight, is assuming all the conditions are the same, the slower the rate of diffusion is going to be. So does that make sense? I don't know if you could see it even. I realized I was sharing the screen the whole time, so you didn't necessarily see the flicking all that large. But are there any questions about that?
Wait, so the lower the weight, the higher the, or the lower the weight, the higher the diffusion rate?
Exactly,
Okay.
exactly. Right.
It's inverse, one to each other.
Say that, Maria?
They're inverse.
They're inverse, exactly. They're
Okay.
inverse. And that's why it's on the bottom here. When this goes up, this goes down. When this goes down, this goes up. So
Could you also say because if it's a higher molecular weight, it's bigger, so then it's less likely to cross the membrane?
no, that's something, that's in fact, that's something in addition. You know, whether or not the permeability is going to depend on lots of things, but not necessarily, it probably is, it could be related, but that's not what we're looking at here. Here we're talking purely the mass of the molecule is the only thing we're looking at. All right.
So the diffusion, are we talking in terms of whether or not if there's a membrane, a semi-permeable membrane in between, or just freely flowing?
Either way, I mean, if we're talking, when we're talking about permeability, obviously we're talking about some membrane in between. So a lot of times we're talking about diffusion across like a cell membrane or something. But if you're just talking about, you know, inhomogeneous distribution of solute in some liquid, you know, then this will not be relevant. Right,
Well,
so not all of these variables will necessarily be used if you're just looking at, oh, here's more dissolved here and less here, then we don't have to worry about permeability. You know, it's like, yeah, I wouldn't worry. Yeah.
will
So
the
permeability is when we're thinking about moving across some kind of membrane. There is this is related. This isn't exactly how you write it, but Fick's law is basically an equation that kind of brings all these in and kind of looks at the rate of diffusion and how it's affected by these things. So this isn't exactly how you'd write Fick's law, but when it does have the basic relationships that Fick's law describes. So if you ever see this word Fick's law, it's basically the equation people use to try to look at diffusion rates that are affected by concentration gradient, about surface area permeability and stuff like that. So, all right.
Excuse me, I have a question.
Uh-huh.
When you say surface area, is that of the screen or the surface area of the molecule, like the solute?
No, of the screen.
Okay.
Of the screen, right? Because that's why, like in your digestive tract, right? If I have my cells here, again, you're gonna have your bloodstream here. You've got your nutrient molecules here, and you have to bring them across. If I have this situation, I have a certain rate of diffusion. If I change this now and I make these with these much higher surface area, now I have way more places where things can cross all in parallel at the same time. That's gonna give me an increased rate that I can bring things across.
Thank you.
Sure. Okie dokie. So let's talk about cells now. The basic unit of life. And I'm assuming that you all have a basic understanding of cells from Bio 110. I am not going to go into the details of all the different parts and pieces of cells. Instead, we're gonna kind of develop the things that we need to look at in more depth for our class. Obviously, there's the nucleus of the cell. What do we find in the nucleus of the cell?
DNA.
That's where you find the DNA. You know, and again, every cell has about 2 meters of DNA in it. If you unwrap that one little molecule, it literally is like 6 feet long. It's a lot of information in there. There's also different proteins, the histone proteins, it's all wrapped around. What's the function of the DNA in there?
Store genetic information.
So it's storing information, you know, particularly about the
Protein.
the primary structure of
Synthesis.
your proteins. It's telling you what the primary structure of your protein, which amino acid to hook up in which order. It's the codons in the DNA. You know, the other thing that the DNA is doing, there's lots of regulation, you know, that telling the cell when it should divide or when it should sit around, what it should make from its blueprint, right? Every cell has a blueprint pretty much to make anything. You don't want a cell in your nose making digestive enzymes so your nose dissolves off your face, you know, or you don't want the cells in your eyeball making hair so you have like hair growing inside your eyeball or something, right? So there's a lot of regulation as well of how the cell functions and what protein synthesis happens and all of that, which enzymes are getting made or not made. So the nucleus is really important, although we're, you know, in our class, we'll just refer to it every once in a while as necessary. Cell membrane, we're going to spend lots of time at, also called the plasma membrane, also called the plasmalemma. These are all just synonyms, AKA, also known as. Again, we talked very briefly last class about what is the primary components that make up the cell membrane?
Phospholipid bilayer.
Say it again.
A phospholipid bilayer.
Exactly, the phospholipid bilayer. And
Triglycerides, glycerides, and
so not so much the triglycerides, but the cholesterol. There's cholesterol stuck in there, in between the tails. What other kind of things do we find in there?
Proteins.
A lot of plasma proteins. We're going to be looking at all sorts of plasma proteins today. We're going to look at all the different roles they play and how important they are in holding cells together and having cells control what comes in and out, having cells sense their environment, having cells communicate with other cells so you can have trillions of cells that all function as a single organism. That's a lot of that's going to come down to these different membrane proteins and all the roles that they play. And then we have obviously the cytoplasm, the stuff inside. Again, we're going to come back, spend a lot of time here at the cell membrane, but let's just cytoplasm, which is basically cytosol, you know, and organelles. Cytosol just means kind of the fluid in there. Organelles are the little structures you find in there. You know, when we talk about, like, you know, glycolysis, the beginning of breaking down cellular respiration, those enzymes are just floating around in the cytosol, and the initial stages of the metabolic pathway are just going to be happening in the cytosol. When we say the word cytosol, it just means the fluid generally inside the cell here. Organelles, on the other hand, are like the specific little things. Like what's the classic little organelle where the rest of cellular respiration happens?
Mitochondria.
Mitochondria.
Yeah, the mitochondria. That's where we're gonna be making the lion's share of the ATP. what do we call the systems of membrane where things can get kind of stored and transported around inside the cell?
Endoplasmic reticulum.
Exactly, the endoplasmic reticulum. What do we often find kind of studded on top of the endoplasmic reticulum?
Ribosomes.
There's often ribosomes on it. In which case it's going to be rough ER, the rough endoplasmic reticulum. What happens at a ribosome?
Protein synthesis.
Yeah, that's the protein synthesis. Remember, that's where the messenger RNA docks, and then the transfer RNA matches the codon and assembles the amino acids. I know for sure you've seen this in Bio 110. You use the little felt blocks and kind of model what happens here. So ribosomes are where we're actually assembling the proteins. It's kind of nice when they're sitting on the ER, the endoplasmic reticulum. Basically, as the protein is assembled, it's just getting threaded into the ER. So now it's already stored and in these system of channels to be moved around where it needs to go. You can also have free ribosomes that are not associated with the endoplasmic reticulum. So those can be, so ribosomes can either be part of the rough ER or they can be free. What's another important organelle that we find in the cell?
The mitochondria.
Yeah, that was that dude we talked about.
Golgi apparatus.
Golgi apparatus, that's another good one. So that's kind of similar to the endoplasmic reticulum in that it's membrane organelle, but it's kind of, people usually it's drawn, it looks like a stack of pancakes or something. And remember I talked about how a lot of molecules are not just pure proteins or pure this or that. So think about these glycolipids. Glycolipids are the main, not glycoproteins. Think about the glycoproteins that are the main membrane proteins. A glycoprotein, it's a protein, but then you've got to do some post-processing in order to actually add on the sugar groups to the protein. So we can make a protein, and then we have to, it's basically like this little assembly line. As it goes from one to the next of these little membrane sacs, there are enzymes that are doing the appropriate reactions to modify the molecule to make it into what you want it to be. So it's kind of like a little assembly line. And then these little things can transport and move things around. So it's a way to assemble things, modify things, and transport things. So Golgi apparatus is important. Other things in there, the lysosomes, if you all remember lysosomes. How do you spell it? Yeah, I think like that. Lysosome, the little things that are filled with digestive enzymes so you can break things down, recycle things. You know, and cells are really dynamic. The pictures we draw of cells kind of look like this usually, or if you look even at a fancier picture, it's still static. There have been incredible advances in imaging in the last couple of years. I went to a talk, this was like about 6 months ago, so we could still go to conferences, where this guy who has pioneered visualizing things in living cells was showing these movies of what it looks like to look inside a cell that's actually doing its thing. It's freaking crazy. Everything is just moving. The mitochondria are wiggling all over. All these little membranes are just pinching off and moving and getting pulled this way and that way. It's so frenetic. like what's going on inside a cell. It's like, imagine like this crazy like factory filled with like hyper workers just doing all sorts of things, and some conveyor belts running everywhere. So the reality of what's going on in a cell is very dynamic. There's lots of stuff going on, things moving in and out, things getting pumped, things getting transported, things getting dragged here and there. So just kind of giving you a sense, cells are very, very alive and dynamic. I should, yeah, maybe what I'll do, I'll see if I can find some like videos of these things that he showed, because even though I knew in my mind that cells are dynamic and animated, like actually seeing the imagery was way cooler than I ever imagined. So, you know, for the purposes of our class, you are responsible for knowing that, you know, what is a ribosome? You are responsible for knowing like what is the ER, what is Golgi, what is going on in the nucleus. You know, yeah, that could be on an exam test. You know, you should know the basics there. The stuff we're going to look at in much more detail are going to be like the mitochondria. So that's something, you know, we are going to go into in this class. We'll spend an entire day just going down, drilling down deep into the mitochondria and looking at all the processes going on in there. And what exactly does it mean to be the powerhouse of the cell? And again, I said the cell membrane, we are going to be spending a lot of time looking at what's going on here at the cell membrane. We've already talked about what kind of molecules can cross the cell membrane without any help at all.
Non-polar molecules.
Exactly.
Small.
Non-polar. So non-polar things can cross easily, and some polar, usually as long as they're really small. You know, everything else, in fact, you know, is going to need help. And again, we're going to be finding all these special membrane proteins that are going to help move things across. So what we're going to do now is go to the cell membrane and talk about membrane proteins. So right, so we know we have this phospholipid bilayer, and then we also know that there are these proteins that are embedded in it. You know, and some of them go all the way through. They have, this is the cytosol. This is the inside, this is outside. Sometimes they are only part way through, right? If you have a, remember the cell is this phospholipid bilayer. So I can have a membrane protein that does this, and it'll stay in place, assuming that the two ends are polar and the inside is non-polar. Then it's going to want to stick in that situation there. But at the same time, I can have something that just sits partway. You know, if this is polar, but that's non-polar. So you can have membrane proteins that are just on one or the other side of the membrane and stay there, and other ones that are transmembrane go all the way across. These are the ones that are going to obviously be used if we're going to have like a channel, you know, if we're going to transport, let things travel from one side to the other. Some of these other ones like this tend to be more like enzymes, things that are catalyzing reactions that have to happen there, but there's no reason for them to go all the way through the membrane. All right. So what are some of the roles that membrane proteins play? There's lots of them.
It can be hydrophilic and hydrophobic at the same time.
Transport.
So one big thing is transport. Yeah. So transport. This means helping things get in and out of the cell. And there are a few different versions of these, and you need to kind of be able to understand them. So one kind are what we just call channels. Channel is kind of like a tunnel that lets things move across. These are typically for ions. Typically for specific types of ions. You know, for example, it could be a potassium channel or a sodium channel or a calcium channel or a chloride channel, or sometimes it could be a divalent cation channel, like magnesium or calcium can go through. So typically it's, you know, and sometimes they can be more generic. There's also leak channels that just let pretty much anything through. So they're not always, that's why I'm saying typically, not always. There are leak channels that can that let in varieties. But in general, we'll talk about channels like sodium channel, which means it only lets sodium through, or potassium channel, it only lets potassium, or sometimes it's just a cation channel, which means it just lets cations through, but not anions. These can be gated. This is going to be super important. So sometimes they're just open, and they're just open all the time, and things will move based on their concentration gradient, right? If this thing is just open, let's say if this was a potassium channel and if there's a lot of potassium, all else being equal, the potassium is going to want to move from high to low concentration. A gated channel is a channel that has a gate on it, meaning that there's something that can open or close it. So these can be open or closed. It's like a door, right? Like the door to your house is a gated opening. If the door is open, people can move in and out of your house. If the door is closed, they can walk up to your house, but they'll break their nose on your door if they try to run through. So gated channels, opened or closed. What can control gated channels? This is going to be important in this class.
Size, ions, or charge.
So that's more going to determine which thing can go through if
Okay.
it's open, right? So those things are important, but those are more determining what specific kind of channel is it? Which one of these things actually gets to go through if it is open? But here we're talking more about the door. Like what is the thing that can open or close the door that provides access to the channel?
Is it voltage?
So one thing is voltage. Which means we also have to introduce the idea of voltage. So we're going to do a little tangent here and describe what the heck is voltage. All right. So tangent. Voltage. Voltage, which is a fancy word named after this dude Volta, for electric potential. Sometimes used with a V, for volt. Sometimes we use an E to describe as a letter to represent voltage, kind of, for electromotive force. Sometimes you'll see this word, the letter E representing voltage, and you're like, why is an E for volt? It's because this, through history, was accepted as a thing for voltage. So let's talk about what is voltage. So let's look at a cell. In general, a cell is going to have kind of charge. There's ions. We have cations like sodiums and potassiums. There's also sodiums and potassiums and other cations outside. And there's anions. There's little chlorides and stuff. So as long as you have a balance of charge on either side of the membrane, if there is just as much plus and minus on one side as on the other side of the membrane, if you take some ion, let me take let's take a potassium here. Here's a potassium kind of floating, or normally if I'm in the classroom, I can actually have a thing I'm floating around in front of the board, but I can't do that now. So does this potassium want to move in or out of the cell based on the electric field, the electric charge?
No.
No, if it's totally balanced on either side of the membrane, there is no potential energy driving the movement of this ion in or out of the cell. There's no force moving it one way or the other because everything's balanced. It's just as likely to want to go in or out because it's the same balance of charge inside and outside. So this would be like no voltage here, or the voltage would be zero. So voltage is zero is if there is no charge imbalance on either side of the membrane. So now we're going to let's look at separation of charge. So now I'm going to look at my cell here. Except I am going to, maybe I'll use a pump or who knows what. I'm going to start pushing some cations and leaving the anions behind. So at this point here, I've started pumping stuff out, and there are more, if you look at just this membrane here, this side actually has more positive, this side has more negative. So I have a separation of charge that has happened here. And again, it could happen if you're pumping things. It can actually happen through diffusion. We're going to look at different things that cause this separation of charge. But this now is going to create an electric field. If I take my potassium again here, and I ask it, which way does it want to go? Does it want to move into the cell or out of the cell? Which way does it want to go?
Into the cell.
It wants to move into the cell because opposites attract. It's positive, so it wants to go into the inside where things are more negative. So in this case, we have a voltage across the membrane. You usually do it like this. So here we're going to have what this would be a negative voltage, a negative membrane potential. if it's always going to be voltage of the membrane is always going to be inside with respect to the outside. So this is important. If I say my V sub n equals minus 70 millivolts, that would mean that the inside is more negative than the outside by a certain amount. And that means there's a certain amount of force that is pulling this cation into the cell where it's more negative. Now, if I have a chloride ion, what would a chloride ion want to do if I have a membrane voltage of minus 70?
Go outside the cell.
It's going to want to leave the cell. It wants to go to where it's more positive. If the cell is minus 70 millivolts, that means it's more positive outside, so the chloride would want to leave. This kind of behavior is going to be critical when we start looking at cell signaling in neurons and muscles and things like that. So electric potential or voltage, it's basically separation of charge across the membrane, which makes charged things like ions want to move in or out based on which side is more positive or negative. So are there any questions about what is membrane voltage? So to bring it back to gated channels. So remember we're saying gated channels. So channels that you can open or close like a door. You can have, one type we said are voltage gated channels. So typically the way these work, cell at rest is around cell membrane voltage is maybe minus 70 millivolts. And the channel will open when VM goes up to minus 55 millivolts, for instance. So this is just an example. And this is an example we're going to see. It's not always these numbers, but this is giving you a sense of how a voltage gated channel works. They like this, like the sodium and potassium channels that we're going to be looking at during action potentials. They're going to be closed when the cell is at rest at minus 70 millivolts. If the membrane potential goes up above minus 55 millivolts, they'll open up. And then if it drops down again to here, they'll close again. So
Doctor, did you say minus 50 or minus 55? Because I think you're saying minus 50, but you wrote minus 55.
Minus 55 is the typical, for the ones that we're going to be seeing, it's going to be minus 55. There are other versions of these as well. This is a kind of a typical, a classic one that we're going to look at in more detail are going to be voltage gated channels that are closed at minus 70 and will open if the voltage rises to minus 55 or above. These are going to be critical when we look at how neurons and muscles are excitable with action potentials. These are going to be critical when we look at even how neurotransmitter is released at the end of an axon when the action potential reaches the end of the axon. So these will be important. What's another thing that can gate a channel, can open or close it?
Ions?
Again, the ions are the things that are moving through the channel. So I'm thinking about what are the things that are going to open or close the door to allow the ions through.
Okay.
Pressure.
So pressure, so mechanical. So you can be mechanically gated. So basically a kind of stretch of the membrane. So this is how pressure receptors and touch receptors work. You know, by this physical deformation of the cell that's actually stretching out the membrane, it opens channels. And when it opens channels, ions can cross, and those ions create an electrical signal that goes to your brain saying, I'm being touched. This is how you're hearing me right now. Your eardrum is vibrating, and that vibration is ultimately working these mechanically gated channels that open or close based upon the vibration, the movement of your eardrum. And that is going to create flows of ions across the membrane, which again, an electrical signal going to your brain. So mechanically gated channels are going to be responsible for touch. They're responsible for hearing. They're responsible for all sorts of stuff like that. So that's another way we can gate a channel, is just physical. This is just a physical stretch of the membrane, opens the channel. When the channel opens, you know, ions can cross, and when ions move, if you remember from the first week, electrical current is just moving charge. So as soon as we have ions moving across the cell membrane due to an opened channel, that's an electric current. That's signaling in your nervous system in a lot of cases. What else can gate channels?
Light.
Chemicals?
So light, actually light can, and we're going to get to that one. I'm not going to write it down here because it's such kind of a different kind of thing. So chemicals, let's say chemicals. We call ligands. So this word ligand is going to be important. Ligand is just the word we use if there's some kind of a signaling molecule that is creating some effect at the cell. So examples of ligand-gated channels would be like the channels that are responding to neurotransmitters. You release a neurotransmitter at the synapse. That neurotransmitter then binds and can open up a ligand-gated channel to allow some effect at the target. Ligand-gated channels, that's how, you know, it's why you get stoned when you smoke pot, because that THC goes into your nervous system and is opening up different channels, creating effects in your nervous system. Ligand-gated channels are also how you detect just the levels of glucose in your blood. Like, is your glucose level right? You have little channels that can detect sugar, for instance. Is how your taste buds work. How do I know that that stuff is sweet that I'm chewing up? It's because I have channels that are opening up in response to glucose binding in my taste buds. So, you know, and creating an electrical signal, which then goes to my brain and tells me that I'm experiencing sweetness in my mouth. So you can have, we're going to have a lot of examples of channels that are controlled by some molecular messenger, some molecular signal, some ligand, some chemical.
So those ligand, the example of it would be like CCAMP, CGMP, AD.
No, I mean, those, they can be, yeah, those are second messengers that can act as ligands. Ligands are much more generic. It's basically anything that, any kind of chemical or molecule that kind of fits and binds with something and has some effect. So I would say ligand is a very generic kind of term. And some of them are internal to you. Some of them are coming from the outside. We're going to talk about exogenous and endogenous ligands in a little bit. So ligands could be neurotransmitters. Ligands could be, again, some drug that you're taking in that's binding and having some effect on your cells by binding things in there. So we'll talk about lots and lots of ligands. So gated channels, voltage gated channels, mechanically gated channels, ligand or chemically gated channels. These are all going to be key players in the body. And this is how cells are going to be sending messages. This is how chemically gated channels, or how cells are going to be just detecting, and having responses to things happening in their environment. What's going on out there? What's floating out around me? Or is somebody sending me a message? So gated channels. So there we've got we're still in transport. We've still got quite a bit more to deal with transport here. We've got transport, we have channels, typically ions are moving. There are going to also be others, water channels we'll see in the kidney, the aquaporins. Most of the ones we're going to look at are going to be more for specific ions. Sometimes they're just always open. Sometimes they're gated, and like then we just talked about 3 things. They can be gated by your chemicals, they can be gated by just mechanical changes like stretch. They can be gated by voltage changes across the membrane. For things that are more complicated than just like an ion, if you start wanting to move like sugar, obviously cells are going to want to move sugar across the membrane, or amino acids across the membrane. Digesting your food, and you got to bring these things into the cells. Those can also cross the membrane using special membrane proteins, but they're kind of more complicated than channels. And these are going to be called carriers. So let's look at carriers. So this is still a transport protein, things that help move things in and out of a cell. So these are usually for larger things. So these are like, for monosaccharides, amino acids. And rather than being just a tunnel, like the carriers are going to be more like a revolving door. So for instance, let's say you have a glucose. It doesn't just go through a tunnel and end up on the other side. It kind of goes here, and then this thing kind of has this conformational change and kind of spits it out the other side. And if I want to go the other way, you can go the other way. But again, it's this conformational change. This thing binds here, and then this thing makes some little movement and it spits out the other side. So things will still move along their diffusion gradient. Things move from high to low concentration, but there is a difference. Like, so this is more, I'm going to say, okay, normally we'd be in person and I could kind of show this to you more, like a revolving door. So imagine if you just have a few people. Does everybody know what a revolving door is? It's just like, here's a revolving door, and here's some people on one side, and you want to go out, you got to go in and and then you're outside. If you want to come in, you go and then you're on the inside. But there's this thing, this engaging. You got to go and rotate around in the revolving door, which means if it's just one or two people, it's not a big deal. They go inside or outside pretty much at the speed they want. But if you start having a lot of people all wanting to go out at once, you're going to get backed up. There's only a maximum amount of people that you can move in or out of this room, because the revolving door has a limited capacity of people that can fit in at any moment and rotate around and end up on the other side. So when you have carriers, because they act more like a revolving door, you end up getting a limit to how fast things can go across. So this is going to be important. So let me show this. If I have concentration gradient on my x-axis, and this is going to be, so is 0 meaning there's no difference between the two sides. Higher and higher meaning that there's a larger and larger difference between the two sides. And then here I look at diffusion rate. So for regular diffusion, what does this relationship look like?
Higher the concentration, higher the rate.
Yes, direct.
Exactly. So this is just regular diffusion. Regular diffusion, the higher the gradient, the higher the rate. That's kind of how we started talking about everything this morning, right? This is going to be something you're going to see in your lab in a little later today. If you are using a carrier, then you get something else that happens. It starts like this. But then I talked about there's a maximum rate that things can move through that revolving door. There's going to be a maximum rate. So does that make sense? So this, we're going to call this facilitated diffusion. Facilitated, wait, I think I misspelled that. Facilitated, no, facilitated diffusion. So if you're using a carrier, you hit a maximum rate that things can cross the membrane using the carrier. And this is going to actually be important to keep track of. You know, it's going to be when we start talking about, much later in the semester, we're going to be looking at the kidneys. We're going to say, how come you have sugar in your urine if you have diabetes? It's going to be all about having a rate limit with facilitated diffusion. We have the carriers in the tubules of the kidneys and the nephrons that are recovering the glucose from the filtrate. And normally they have the capacity to pull it all back, but when you have diabetes, you overwhelm their ability to pull things back since they're rate limited, and you end up having, glucose spilling out into the urine, et cetera. And when we talk, there's a variety of reasons why you actually do need to know that if we are using carriers to move things across the membrane, that there is a maximum rate that you hit at how fast things can actually move from one side to the other. For normal diffusion, you don't have to think about that. You just think the higher the gradient, the faster things go. But if you're dealing with carriers, that's why we give it this special name, facilitated diffusion. It's still passive. this is still passive transport. It's not taking extra energy to make it run or anything. It's just that we have this additional thing of it being rate limited. If you're using a carrier to move things across the membrane, again, you need to remember you might hit this wall where there's a maximum rate things can use the carrier to cross. So any questions about what we mean by facilitated diffusion?
So both carrier and the channels, they have a passive transport, you're saying?
Yeah, assuming that you're just doing diffusion, it's passive transport. And we're going to be seeing in a few moments other kinds of transport proteins that actually are not going to be passive. We're going to look at different forms of active transport. But if you're just opening like a gated channel and you open it up, assuming nothing else is going on and things are just moving according to their concentration gradient, that would be passive transport.
Okay.
Okie dokie. All right, so carriers, like a revolving door, things move in or out. Now we're going to get even kind of more complicated, these co-transporters. Co-transporters, these couple the movement of two or more molecules at the same time. These can be symports or antiports. A symport would, an example of a symport it's like glucose can come in, but only if it's coming in along with sodium. So we're going to see one of these in a moment. An antiport means that things move in opposite directions. We'll see a number of antiports that are important. Like, potassium moves in, but only if sodium moves out at the same time. So these are coupling co-transporters. So coupled movements.
So is it like only two at a time or more than two?
It depends which one. Like one of the main sodium potassium pump is actually moving multiple sodiums and potassiums at the same time. So it depends on the particular one you're looking at.
Is it true with the symport as well? Like doesn't have.
Again, I think you just look at each one as individual. Right now we're just talking about the more general description. Sometimes they move things in together, sometimes they move things opposite directions. And then we'll see there's lots of different versions of these with different characteristics. But this is the most broad description of them.
Thank
you. So co-transporters, and then the last thing we can also talk about, there's things that kind of act as pumps. So these actually can like use ATP to move. In fact, let me get a new page. Let's talk about active transport now. So far, everything we've been talking about is passive. You're opening up channels, you're letting things, or you're having carriers and things move one side to the other side. Even these co-transporters, coupled transport, there's still, things are moving to one side or the other side based on concentration gradients. Active transport is gonna use energy, as opposed to passive transport, which does not need any additional energy. typically, moves things from low to high concentration. Against the concentration gradient. So this is if there's already a whole bunch of sugar in the cell and you want to pull even more in. Normally sugar wants to move from high to low, but the cell is saying, I don't want to let the sugar leave here. I want more, pull even more in. You're going to need active transport, something that will pull the glucose upstream. So active transport, there's a few different ways to do this, to run things against their concentration gradient. And we're going to look at those right now. One, we can have what's called primary active transport. This uses ATP directly to pump things. So there's going to be a variety of primary active transport. Like one of the important pumps in a lot of the cells is going to be this thing that pumps sodium out and pumps potassium in. And in order to make that run, you're going to take ATP and break it apart into ADP plus phosphate. So this would be a primary active transport. It's using the energy from ATP here. How do I, there, no, I'm trying to use my little, oh, there it is. Okay. I'm using the power of ATP and breaking it apart into ADP and using that energy to pump the sodium against its gradient and potassium against its gradient. So that's an example. Another one we'll see are like calcium pumps. You're pumping calcium into some, reservoir, and to do it, you're going to have to take ATP, break it apart into ADP plus phosphate. Right? So calcium, let's say there's already a lot of calcium in here, not much here. Calcium does not want to move in, but you can drive it in using the energy of ATP in using this pump. Right? So this idea, primary active transport, you use ATP directly. This is going to be in contrast to secondary active transport, which is going to still move things, still going to use energy, is still going to move things from low to high against their gradient. However, this other part we're going to do now, the secondary active transport, is not going to use ATP directly. So let's see how that works. Does not use ATP directly. I mean, I'm gonna say these words, and they're probably gonna be like, what is he talking about? But it's gonna make more sense when I just show you an actual example. This is something that's way easier to show than say. So basically, I turn on Mr. Keyboard. Oh, dag nabbit. All right, my little keyboard has lost its battery, so I'm just gonna write it out using my pen. So uses energy stored in a concentration gradient along with a co-transporter. To move things against their gradient. So I think the best example of this is something that's going to illustrate a lot of the things we've just been talking about, and also something that you should know is going to come up again and again. It's going to come up in how we absorb nutrients from our digestive system. It's going to come up in how we process the filtrate in our kidneys to adjust the concentration of things in our blood and all that. So we are going to talk about trans-epithelial transport right now. I need to... So trans-epithelial transport. So epithelium, just a sheet of cells that kind of is lining things. So let's think about your intestines. Here's your intestines. Here's all your kind of digesting food, you know. There's some epithelial sheet here. And then you have blood vessels here. So as the food digests, some of it's going to just end up being your poop and continue out the end of the tube. Other things going to be interesting nutrients that you want to pull across into your body. So I'm going to now look at this area here that's particularly intriguing to us now. If you remember from anatomy, I know a lot of you have been in anatomy, what kind of epithelium makes up the lining of your small intestine?
A simple columnar.
Yeah, this is like my simple columnar epithelium. So these are my cells, my little simple columnar cells. You know, this is going to be up here, this is going to be the lumen of my, lumen meaning the inside, where all the food's digesting. And I'm going to be ending up with these yummy little glucoses. That are the breakdown product of digestion. So I've got glucoses coming into existence during this digestive process. There's other stuff, like I said, that's going to end up being your poop and continuing on. But the glucose are interesting to us. On the other side, in the lamina propria, we're going to have capillaries. That's where we absorb our nutrients to bring it into our bloodstream. So this is the wall of my intestine here. This is my intestinal lining. right? This is the inside of the tube where the food is breaking down, where the sugar is coming into existence that I want to absorb. Here's the capillary on the other side of this epithelial sheet that I want to bring the sugar over to so I can get it into my bloodstream. So trans, meaning across epithelial transport. I want to take these sugars, which are in the lumen, and I need to bring them across to the other side of the epithelial sheet so they can get absorbed into my capillary. And I'm going to talk about how that happens using these coke transporters and pumps and all this stuff that we've been talking about. So let me lay it out here. One thing we're going to have is we're going to have a sodium pump here. And this is using ATP. So it's just actively pumping sodium out of the cell. So it's making sure that there's more sodium outside than inside, and there's low sodium inside. There's not as much sodium in the cell because we're actively pumping it out. What do I call that kind of active transport if I'm using ATP to pump things?
Active.
Primary active transport.
Exactly. So this is going to be an example here. This is primary active transport. So that is creating a gradient. It's creating a sodium gradient. There's less sodium inside than outside. So now sodium, if it has a chance, it wants to come in. So what I'm going to do is I'm going to have a symport now. That couples the movement of glucose with sodium. So glucose comes in, but only if sodium comes in with it. So this is actually going to be my secondary active transport. Sodium, again, I tend to personify these molecules, even though they're not alive. They don't have wants and desires, but you can think of it like the sodium wants to come in because there's this gradient. So the sodium wants to come in, but it can only come in if it drags a glucose along with it. So this is what I'm talking about when I was talking about the idea of secondary active transport. So the glucose normally would not come in because the glucose, there's already a lot of glucose in the cell. Whoops. So there's lots of glucose in the cell already. But we can bring even more glucose in against its gradient because the sodium gradient is driving the movement using this symport. Does that make sense? So that's secondary active transport. It's using the energy stored in the sodium gradient to drive the movement of glucose against glucose's concentration gradient.
One question, Professor. This is as a result of the gradient created by the pump.
Exactly.
Okay. Okay.
So this secondary active transport would not work if we didn't have this primary active transport creating the gradient in the first place. So they're related that way. But if you look like exactly where is the energy coming to move the glucose, the energy is coming from the sodium gradient. But the sodium gradient came because of the ATP running the sodium pump here. That makes sense. All right, so then we have a bunch of glucose building up in the cell. We still need to get it out the backside of the cell. So on this side, we have lots of glucose just moving into the bloodstream. For here, we can just have a glucose carrier. Because basically, as the glucose ends up on the backside here, it gets taken up into the capillary. So we constantly have a diffusion gradient to move glucose out of the cell. We have constantly have glucose building up in the cell as we take it up on the apical surface on the top here. And it's getting taken away into the bloodstream on the backside. So here we don't need to pump the glucose. It's just going to move according to its diffusion gradient. It's happening using a carrier. So what do we call this? What do we call what's happening here?
Facilitate.
Exactly. This is facilitated diffusion. So here in this example of transepithelial transport, it's illustrating a lot of the important concepts we've been talking about. And like I mentioned, this is going to be happening all over the place, and we're going to see this happening in different systems because controlling the movement of things from one side of an epithelial sheet to another is kind of the critical way you're controlling the environment inside and outside of your body. So, are there questions about this? So this is like one of these things, like I promise you that transepithelial transport is going to be on your exam in a pretty direct way. So let me get the fat. Trying to highlight it here. You know, if you go into the exam and you don't really understand this, you're hurting yourself. You know, so make sure that this all makes sense. This isn't just like, oh, he kind of went through that, now let's see what comes next. If this doesn't make sense, what I just kind of described as happening with the primary active, the secondary active, facilitated diffusion, how we move stuff from the lumen to the capillary side, you know, you want to review it, you can ask questions, figure it out, but make sure you don't let this slide, because this is illustrating a lot of important concepts that, again, that we're going to be finding in different forms as we go through the different organs and systems and stuff. So, comments, questions, otherwise? All right, so it is now 9:40. Let's come back in a half an hour and we'll do the quiz. Again, the quiz is going to be in Proctorio, so you need to make sure you're on a laptop with a camera and stuff like that. And we'll talk a little more about the details of the quiz when we come back. But for right now, and I'll also be open for questions. Are there any last questions before we start the quiz? But otherwise, I'm going to meet you at 10:10, and we will do our quiz one. So I'll see you all in a little bit.